Welcome to Hypothesize That, UCB's science podcast, where we'll confront some of the biggest trends, theories and questions in the world of pharmaceutical research and development. I'm your host, Nicola Hill, and I'm joined today by two very distinguished scientists. I have Dr. Alistair Henry, who's the Chief Scientific Officer at UCB, and joining us virtually from America is Russ Altman, who is Professor of Bioengineering, Genetics, Medicine and Biomedical Data Science at Stanford University. Gentlemen, welcome. I think this is going to be a very interesting discussion we're having today. We're going to be exploring a bold and rather provocative hypothesis. Will digital technologies revolutionise the pharmaceutical industry to the point where traditional wet lab work becomes obsolete? We'll be exploring ideas that tools such as digital twins could reinvent the pharmaceutical industry, and we'll explain how they work, their advanced simulation capabilities and comprehensive data integration. So, pull up a chair as we dissect the future of pharmaceutical research. So I'm going to start with you, Russ. Obviously, our hypothesis today is, will virtual technologies eliminate the need for these wet labs? Is this a reasonable hypothesis?
So the word eliminate is a strong word, as you know, but I would say that we have seen trends towards the reduction in the role of experimental biology, and I think that's something you can count on. What I tell my students and sometimes my colleagues in the pharmaceutical industry is you want to do everything you can in the computer before you move to experiment. And that has not been the tradition in the pharmaceutical industry, where it was go to the lab first, collect the data, and then see what you have. I think the new model will be do as much as you can in silico, and then when you're at your wit's end and you really think you have a winner, then go to the lab and check to see if you're right.
Yeah, that definitely makes sense, but let's just push this a little bit further. Alistair, I mean, let's talk perhaps about some of the digital technologies that we've already got in place and how they're changing the pharmaceutical industry.
The change has been enormous, and it's everything from right at the very beginning of identifying the patient populations you'd want to look at, the right patients, what is the underlying cause of their disease, the pathobiology, what are the mechanisms that drive their disease, and then of course all of our screening activities and generative technologies to make better molecules. Again, digital has had a big impact in all of that. And then, of course, you carry on right through to the far end, toxicology, and then ultimately you do get back to a patient again. So clinical trials, how we recruit, what do those trials look like? And ultimately, the data you get back. How do you understand the data that you've got from very diverse patient populations and then use that to infer the much bigger population? We don't do a trial in every human being. We do a trial in the representatives who then help us to understand the rest. And so again, digital has had a huge element of that. So, and this with Russ, do I think it's only desperation that drives us to the lab? I will gently disagree and say it's the fact that we have now perhaps refined our hypothesis and can make more use of what is actually really expensive time, which is the time in the lab.
I think perhaps I should have asked you this before, gentlemen. Should we explain wet labs? Because I think that's something that people assume that all labs are wet labs, that, that's where the scientist is there mixing his potions in the Petri dishes. Can you elaborate a bit more about what exactly a wet lab is and does?
Wet lab is where you will do a physical experiment with the entities that you're interested in, and ultimately you could even say a wet lab extends to the clinic. It's with real people, real studies. Of course, what we're contrasting is what you would do computationally, which is still experimental. It's still science driven, if you like, but it's not literally with molecules, cells, animals, people in that way. And that's the distinction that we're seeking to make here: computational and that which is not computational in its inherent essence. Would you agree, Russ?
Absolutely. And I think the reason this is important, and thank you for raising this issue, is that these experiments are typically very expensive, both in time and in reagents, that is to say, the chemicals that are used to run the experiment, and in the facilities that are required. And so they're critical, but they're incredibly expensive, whereas electrons moving around in your computer can be much cheaper. That's the appeal here, is drastic savings in time, money, and personnel costs in with hopefully an equivalent result. And we're not there yet, but there's these trends, major new data streams that were not available 20 years ago, electronic health records and biobank records, um, molecular measurements like the genome and other measurements of the cells and the molecules in the body, and now sensing and wearables, both of the environment in general and what your body is doing at the moment. When you have these three streams converging, that's where you begin to get some confidence and some optimism that maybe you can build these models to be very good mirrors of the actual physical world.
I think it's interesting you just said 20 years. I mean, we've gone a long way in a short period of time, and I think that's why some people embrace it and some people are terrified of it.
I remember when I started my PhD, so 30 years or more ago, where there are experiments I do today which I could not do then because we didn't have the computational power, and that was just simply fitting curves, data fitting. Really what is now almost trivial in that sense. We couldn't do multi-parameter fitting because we didn't have the computational power. So that's the shift in 30 years, from struggling to do that to then now actually it's vast compared with where we were, but it's about now creating the additional value for what you actually do in the wet lab.
Absolutely. I totally resonate with that comment, but that has big implications for the workforce because comfort with computation, comfort with digital technology is now not optional, and that changes the types of folks you might be recruiting and also the types of retraining or professional education that you want to develop for your existing staff.
Interestingly, I just saw an advert in a newspaper in the UK. Queen Mary's University in London is actually running an MSc degree course in artificial intelligence for drug discovery. So that obviously is a degree course that 10 years ago would never have existed.
We have seen that our friends in computer science don't always understand enough about biology. And so some of them have now said, I find this to be a fascinating application area. I want to dive all in. So you're having people from the technical fields entering biology, chemistry, pharmaceutical. And then you have the pharmaceutical veterans who are saying, well, this computer stuff looks pretty interesting. And they also have a need for information. And so now we can count on a cohort of young people who will grow up with both of these bodies of knowledge at the same time in the same brain. I understand AI and I understand drug discovery. And that might be the biggest positive that we're looking at in the future.
Okay, let's pick some of the ways that AI is being used in drug discovery. What about digital twins?
We are very interested in digital twins. The high level idea is if you collect a lot of information about an individual, you can put that in a computer and you can use AI to, rather having it be a static data set, you can actually ask it questions like, what if I change this molecule? What if I block this molecule's function? What if I add this drug into the mix? What would happen? And you can do AI simulations that can be predictions of what would happen if you gave that actual person the same perturbation, the same drug, the same change in their molecules. So that is very exciting because now it's individual. All those individual data streams and all their special differences across the population can be captured. And then you can now do trials on your drug before you ever put it in a patient. You can simulate what it might happen to that patient and to thousands of patients, and then have some more confidence, as I was saying before, to then bite the bullet and actually give it to those patients. But obviously that's a big deal, and so our digital twin technology needs to improve to the level where we really believe that those results are sufficiently accurate to act.
Yeah, there must be caution, I imagine, Alistair.
So I think there's two elements for this. So one is a confidence thing, and that is that, of course, there are parts of that modelling that Russ has described where we do know in real detail the parameters, so the mathematical modelling, which is something we've done for a long while now, to make those predictions, and there's robustness in that. So we have confidence in the one hand. But the cautionary note is, it assumes that I actually fully understand the whole organism biology. So the interplay of all these systems and the interplay of my molecules, my new medicines, in those systems. And of course, one of the big things that one has to really grapple with is that not every molecule you make has all the desired pharmacology that you want. Some of it has some adverse pharmacology. We call it toxicology. So, and if I could predict all the toxicology, then I would ultimately not have any tox, because I could predict and remove it. But that's the problem. We don't know fully how these systems completely interact. So I think there's a great benefit of the digital twin, but there is a cautionary note, because we need to acknowledge we do not fully understand the biology of the systems that we are trying to manipulate.
And indeed, I completely agree. And if I can just add a friendly addendum, which is that, and that is the reason when drugs fail, it's very often because there was a big hole in our understanding of biology that we either knew about, but we couldn't do anything about because we just didn't have the knowledge, or we didn't even know what we didn't know. And so fully elaborating the biology of health and disease is still a big goal for the field.
I actually was reading something this week about, they reckon there's a gentlemen, you can correct me, this dark mass of compounds that we don't even know exist at the moment. Is that right? And the AI is going to help find them.
Now we're into the definitions of chemical space, and what's the total number of molecules you could ever make with it? Could the universe ever contain them? And could you ever elaborate them all? And could we ever then define for them all what their properties would be? And the last 10 years have been full of predictions of how big chemical space really is. Or in fact, or do you actually need to explore all of it? Can you actually condense it down to smaller elements of those molecules and find out their properties and then bolt it all back together? So now you're halfway through your MSc in drug discovery, and these are two strategies. Do you start with every molecule that you could have, or do you start with all the fragments that you could build back together to create the molecule? Either way, the numbers are big, and you have to have a way of reducing those numbers so it's practical and ultimately attainable to put a medicine on the market.
The other part of the darkness, so to speak, is that, we sequenced the first human genome about 20 years ago. We have about 20,000 genes plus variations. There are many genes in the human genome that we have not assigned a function to. So this is another type of dark matter, which is the set of proteins in the body, which, as Alistair just said, could be the source of toxicological responses. And so a urgent need in basic biology is simply to label the function of every gene in every context so that we have some ability to predict how they might impact drug response or disease processes.
So another advantage of AI. I mean, we're selling it here, gentlemen, aren't we?
Well, so I'm going to slightly try and unsell it and then resell it for you, right? Repackage it for you. So just to add to the complexity. And so for every protein, we might not have yet fully articulated all of its functions. But let's be clear, what we have also not done is fully articulated all of its states, its conformations, its shapes. So we have molecules that can shapeshift. And so they have different functions with different shapes. And your pharmacology, the effect of your drug, is going to be how it affects those different shape states. Now, if we're struggling to understand the genome fully, I can tell you now we're definitely struggling to fully understand the conformational space of the proteome, the proteins that make up the organism, the human. And of course, AI can help us there because it can help predict those states. But remember, we don't know them all. So we are now in a world of prediction and working out which of those predictions is best and most helpful to drive drug discovery.
I was also reading, now you will be able to correct me if I'm wrong on this, Alistair, but the cost of a drugs and how long it takes for traditional targeted drug development, large numbers. So presumably AI is actually going to help research and development, reducing both of those.
Yeah, I mean, cost and time, which are of course related. If we were able to get there quicker, to make fewer molecules along the way, to have more meaningful experiments, more experiments that don't fail, if you like, then of course that will have a direct impact on both of those components. And as we've said already, experimental time in the lab is expensive, and time in the clinic doing clinical trials is even more expensive. So you want to limit those as much as you can, but get as much information back as you possibly could have.
You know, our last few minutes have been focusing on the great challenges and promises of molecular level information. We've been talking about small molecule drugs and proteins, but very early, Alistair said something very important, which is that AI is also helping in selecting the patients who should be in trials. Identify me the set of patients who have the same disease and for whom it would be useful to test this new drug, and that could really cut the costs of the clinical trials, which is where the lion's share of the $1 billion or whatever number you want to, or £1 billion that you want to use to characterize the cost of drug development. Those clinical trials are very expensive, and AI is amazingly good at finding patients that look alike, patients that are different. It can cluster them based on shared features. That will be very useful for identifying patients for recruitment into trials, retaining them in trials based on their experiences, and then ultimately educating them about why they and their physician should be using these drugs. So the AI's communication and patient analysis capabilities should not be underestimated here.
That's quite important with immunotherapy, isn't it?
It's for all conditions. I mean, not just the ones that make the headlines. I mean, we're a very diverse population. I mean, we all look different. There's a reason we are different. We're genetically different. And those differences manifest themselves even in common diseases. So there is no such thing as a typical patient. And we need to understand that. And that's where this can help us. And really gives an insight into segregating patients into the right groups. So not only can we put the right ones in the right trials, we put the right ones on the right medicine. And if AI can help us get there quicker, absolutely.
Okay, gentlemen, I'm going to challenge our hypothesis now, because I think it's important to consider the other side. One thing in particular that a lot of people criticise AI about is the fact that it can only work with the information that's been inputted. Basically, it doesn't know the unknown, and therefore it has limitations and it has ethical considerations because of that.
Yes, so it's absolutely true that we train these models with the data that is at hand, at least currently. And that means there are certain things we know how to measure very well, and we measure them, like the genome. We can sequence DNA now extremely efficiently, quickly, accurately. So we use that information. There are other similar things that we can do. Unfortunately, there are certain important types of information that are still very hard to measure and to gather. And so our AI models are a little bit invisible on those types of data, and yet they're important for biology. So we have to always be aware of the biases, and I'm going to use this word bias in several senses. The first bias is the scientific biases that might be present because you're not able to provide it all the data it might want to have just because of the limitations of our current capabilities in the lab. The other kind of bias, all the way at the other extreme, is bias because patients are different. Alistair just reminded us that our genetics are different and our environmental exposures are different. And if you don't carefully consider that, you might wind up giving a patient the wrong medication because you've made assumptions about that patient and their background and their biology that are not true. And we have examples of this, especially in underserved populations, where they don't always get the best treatments because we don't have the best data about them. So bias is a big problem with AI that the community is aware of. I don't think it's only technical solutions. There are technical people working on eliminating bias from algorithms. I think we also have to think about it as a social mandate to make sure that at all scales, these algorithms are fair.
Yeah, I was reading about in the UK transplant system, the way that the algorithms have been designed. Anybody under the age of 40 is far less likely to be able to get a liver transplant because AI has decided that they're younger, therefore they'll live longer, so they're not prioritised. So this is an example of the bias.
And it is, and that can play in both ways, of course. We should be very conscious of that. Of course, if it means that we truly prioritise those who have need, and I'm not speaking about specifically this example, but that's a good thing to make sure that we do prioritise, because we know the healthcare budget is limited. That no country has total mandate for everything. So choices have to be made. And if it allows us to prioritise well and to make sure that our biases, therefore, are about those with most need rather than some other function, that is also a potential benefit for us. But it is something we have to know that we have done that, and therefore be comfortable that we can justify that we've done that.
It also raises the issue of explainable AI, where if the physician and the patient who've been told that the patient is a lower priority they might want to say, tell me what the AI used to make this decision. And that's not an unreasonable request. Some of these algorithms, however, are not very good at explaining how they came to the decision. And that can be very troublesome, especially when human lives are literally at stake. So there is a big area of investigation amongst computer scientists on how do we take these complicated AI models and boil them down so people can understand how they made the decision.
Can I just build on that for a moment? Of course, physicians have been using algorithms to make decisions about patient care for many years. So we're already down that road of making decisions on this sort of basis. Now it's to explain it to the next level. And as the reason Russ has said, if you can't explain it, how can you justify it? And if you can't justify it, then you're going to find yourself litigated, I suspect.
Let me quote the godfather of AI, Geoffrey Hinton, the British Canadian who just won the Nobel Prize for Physics. And he's raised fears that humankind may soon be outsmarted and outcompeted by its own creation. And I'm going to quote him directly. He said at the press conference when he was given the prize, "We have no experience of what it's like to have things smarter than us. It's going to be wonderful in many respects in areas like healthcare, but we also have to worry about a number of possible bad consequences, particularly the threat of these things getting out of control." Now, are you concerned about things getting out of control and of these machines outsmarting us?
No, that's a straight answer for you. But let me elaborate a bit further. So, am I worried about things being smarter than me? Well, actually, that's probably most of my colleagues. So I live in an environment, we work in environments where I'm actually really glad I have people that are smarter than me, because I can build on their knowledge and build on their experiences. So that piece I'm not worried about. Does it mean that I might therefore do different things with my creativity? I have no problem with that either. That's the long history of mankind that has done different things with its creativity as its technologies have evolved. So again, I see them as opportunity creating rather than a fundamental threat.
I totally agree with Alistair. First of all, I am not worried. And in fact, the leadership at Stanford recently asked me to lead a committee on this issue of AI and how to make sure it's beneficial for humans and not the source of catastrophe. And there are principles. And if I could just give 3 or 4 of them. One is human oversight of AI. There should always be a human watching AI and making sure that the AI is working as it's intended. And there really shouldn't be a time when it becomes out of control and doing things on its own, because that should never have been allowed in the system design, A. B, human alignment with human values. We have to build these AI systems so that they do things that we want them to do. And we can build that into their algorithms. So they have to align with things that we or our institutions care about. And then professionalism. Humans have to take responsibility for the AI that they use. I tell my students and everybody else who asks, I never want to hear anybody say, the reason we had this catastrophe is because I trusted AI and AI made the mistake. No, AI doesn't make mistakes. The humans who are taking the output of the AI and then making decisions make the mistakes. And there's a professional responsibility not to abdicate your responsibility and let AI just do things for you. And then, of course, there's issues of ethics that we've already touched upon, and data, control of data, and judicious use of data. So I think that we want to make sure that these kinds of principles become articulated and explicit, and that every AI system is built with them in mind. And if every single system is built safely, then the aggregate set of systems should also be safe. And so I do respectfully disagree with our Nobel Prize colleague.
You mentioned then about sort of people taking responsibility and human oversight. Of course, this is one of the other criticisms against our hypothesis, is the fact that, you know, if we get rid of the traditional wet labs, everything's digitalized, are we going to have staffing problems, or is it, as we alluded to earlier, we're going to have a different type of staffing? Everyone's going to have to be digitally savvy.
I don't worry on the subject of staffing. I think we will end up with a different type of workforce that will speak both languages. We alluded to that right at the very, very beginning. You know, there are now university courses which are spanning the gap between the computational and the biological, if you like. So I see an evolving workforce. Where I think maybe we need to explore perhaps a bit further, though, is knowing that, and knowing that there's the that link between the two, the computational and the digital. How will we address the fundamental question that patients will ask us? They will ask this question: does it work? And is it safe?
Yep.
And the payer will ultimately ask, is it worth it? And unless we can prove that to their satisfaction, we are going to be in trouble. And the only place today we can prove that is in an experiment.
Human trials.
Human trials. And so the question of, is it safe? Does it work? Is it worth it? Are only played out there. And so until we can answer that one, we will always end up in the laboratory again, which as an experimental scientist, I'm very pleased.
I am very comfortable with that. I am not an experimental scientist. The only lab equipment in my laboratory is the espresso maker, but I entirely agree with Alistair, which is that we computer people can do everything we can and should do to contribute to that process. But if you're going to give the drug to real patients, then you better make sure you test it on real patients. We can do everything to make it as safe and efficacious as possible, but in the end, that will not go away. We're just really talking about, I think the meaningful part of this hypothesis is in all that work leading up to that trial, what is the balance of experiment and computation? And that's what will be changing over the next several years.
Because obviously some medicines have been discovered by accident, haven't they? I mean, it's in the wet lab. You've got penicillin or things like, various other ones. If you eliminate them completely, surely chance has been eliminated.
The computational world is very excited about this concept that we call repurposing, where you have a drug that does X, and then you find out by serendipity that it also does Y. It might be more useful doing Y than it was doing X. So that sometimes can happen in clinical trials, but you can also write algorithms to look for these, Alistair referred to these toxicological effects, these side effects that are not expected. And you might say, wow, this is not really a bad side effect. This is a side effect that might open up whole new possibilities.
But the real key piece here is to have an open mind to see what you didn't expect to see. Now, AI could help us do that because it will look at all of the data. So we will be able to plot these things and find relationships that we didn't we hadn't anticipated. But we've got to be open to them to then understand what does that mean and where will that take me.
Gentlemen, I think we could continue this discussion for several hours, actually. There's loads more, but time, as always, is against us. Do you think we've proved our hypothesis? I think we've managed to argue against our challenges fairly well there. Thank you very much for joining us on this Hypothesize That UCB podcast today. Absolutely fascinating. So that's a wrap. As we explored the shift from traditional wet labs to a virtual environment, I want to thank you both again. It was great for you giving up your time to chat to me today and helping to navigate the complexities of digitalisation in pharma research. It's clear there are numerous benefits, certainly to the digital world, but we still have some challenges to overcome. But I hope you will join us again as we discuss other interesting topics about science and speculation of its future on Hypothesize That.